So I'd like to open the forum with some open discussion on the four presentation. So is there any question for any of the speakers? Yes.
Hi. I would like to know about the different techniques we have seen. How do you validate, because regulatory requirements, one of the regulatory requirements is to have your method validated already. Do you do something according to ACH guidelines, for example, ACHQ2, or something about robustness, reproducibility? Do you have a systematic way to validate the techniques, the analytical techniques, in order to compare, maybe?
Yeah, for DLS, they mentioned this morning that there are reference materials that exist.
Yeah, the ISO.
So there are still new materials coming. I think the GRC is working on it as well. So most of the time, people use standard materials to validate the measurements. After that, depending on the complexity of the sample, the measurements can be more or less tricky. So at some point, we know that we do at least as good as standard DLS would do, you know. And so if you have a doubt about the measurements, the only way to lift that doubt is to use other techniques. That's what my experience when you discuss with the people in the lab. If they want a quick test, eventually they would use DLS or another technique. If they really want to give a deep understanding and understand the full properties of their sample, eventually they would use TEM imaging or different techniques.
But according to repetitivity, reproducibility, maybe if you all do the same, we can compare which are more robust or more repetitive.
Yeah. So yeah, but the repeatability of DLS, but I guess it's not only the case for DLS, is really related to the sample complexity. Yeah. You know, if you take a latex, you have a less than 5% repeatability,
if
and
we
it's
use
very good.
Standards, as
yeah,
we
so
are.
that's why you have to be aware of that, and that's why I come back to my comment about the fact that people are poorly educated, and most of the time they use Z average and PDI value. They don't even know that there are several algorithms that they could use in a sample, so they end up with a you always pay back by the effort you put in your analysis.
I think as we discussed this morning, and I also link up to the conversation we had with Hans and Jeff, it's your biological outcome that determine your characterization needs. And in certain cases, you don't need to have an accurate, you just need to have a batch reproducibility in your production line. That has to be validated, so therefore it has to be in compliance with the guidelines. But as I said, and also to reintegrate the discussion, DLS is the entry point with or without the getting into the discussion that we had this morning on the quality and the user specificity, but it has to be a tool that has to be parallel with orthogonal techniques, because it's no longer only acceptable as a single technique from that perspective. So the more evidence is provided, and also this was also resonated by the FDA, the better it is. The more data you provide and good quality data you provide, the better you support your case. And I believe, Santi, you confirmed this aspect into your...
In fact, I wanted to say that here we can use the reference somehow, I don't know if you agree, of the, for example, the pharmacopoeias dealing with laser diffraction, which is a technique covering a wider range. But all these pharmacopoeias are providing some procedures in order to evaluate the repetitivity, the intermediate precision, and it can be implemented as well in the DLS. The issue is that in relation to the acceptation criteria, then this is the point in which you need to take into account which is the material, the kind of material that you are analyzing, and perhaps the acceptance criteria that are proposed in that pharmacopoeias can work or cannot work. But at least it can be a good reference, and in some cases we are using them as reference.
It's a starting point, and then has to be matched with what is your reference aspect or your reference material you want to compare, or a biosimilar or where you produce sick. That's why I was asking about efficacy in the response in this particular case, because that would be the critical point, because it is a met clinical need. There is an evidence that there is a need for reaching, but if it is superior and if it's localized. So that's one aspect. Hans?
On the size, there's a couple of issues. When you say that it may not have efficacy, unless you can measure it and check that, you'll never know.
Yep.
So you need to do the research, you need to know. The other one is a lot of techniques give you a relative size so you can get a particle metrics machine in Austria, a Melbourne machine in England, and something else in the US. They'll all give you different numbers for the same sample. So what you need is the objective actual number.
Yeah.
All of these things are making up a number. It's just a software generated. You've got all this hardware correlation, all that stuff. So, and what I think you need, everybody is the real number. And there's even a problem with hydrodynamic diameter. It's not the real size. So the pharmacopoeia are 10 years behind the times. Sorry, they are using technology from the 1980s. And it's a real problem for the industry to wake up and say, well, sorry, we just want to move beyond that a bit.
This is where the precautionary note that they are actually adopting in the new, for instance, guideline from the FDA, they actually are science evidence-based. So they want to be convinced that the science behind is good and the data is good. Then it doesn't really matter.
And the science behind the old stuff is not good. And that's it's really
no,
quite
but that's
poor.
where they come in with they not accepting any longer one single measurement. They want to have at least one volume, which could be an electron microscopy technique, a dynamic measurement, and if it's necessary, they want to have a third, which could be either a fractionation system or it could be electroporetic or a surface charge. Depending on the outcome and the biological outcome that the product is designed for, they come back by asking this. And I think, speaking from the UNCL perspective, for the cases that we've seen, we analyzed more than 30 cases under our process. We have seen that this is very much driven. I think, Jeff, you can second or you can resonate the aspect, because you guys in NCI, you got more than 340 cases.
Yeah, I mean, really, it comes down to, again, just to reiterate what we talked about this morning, it really just comes down to making multiple measurements using different techniques. I mean, really, that's what it comes down to. And then, like I mentioned this morning, again, you want to link this to some biological outcome. I don't know how else to put it that way. I mean, that's the main point with everything. So.
Mm-hmm.
Yeah, I would like to add, basically, what I followed up in the discussion, the concentration typically is hardly measured since, I don't know, I think it's also lack of standards, to be honest, and also it's a need for particle size and concentration standards, and a measurement technique is only as good as your standard. So the closer the standard to your product, the better it is. I mean, we know a lot from analytical chemistry, where they use, I don't know, if you want to measure gold, you use a special amount of gold. If you want to measure silica as an element, you need an standard for silica. And in particle analysis, it's more a multidimensional problem, because with particles, they are metastable. Some of them are very stable, some not. So what I've learned from experience is each sample is different, because once you capture the chemistry behind and the stability, you know a little bit more. But it's really crucial when you do assessment in terms of quality that each user is doing the exact same thing. So you have an impact of the user as well. And here it comes to standardization and also the quality of the standards. So for us, perhaps you remember one of the slides, we evaluated 4 different concentration size standards from different companies, and we were shocked. So 60% difference. On a log scale, it looks great. But if you put it on a lin scale, you see some interesting results. Again, adding concentration is adding another big challenge. I've been in many DIN and ISO committees. concentration is neglected, and this is really a big thing. There has to be a lot of work to be done. Yeah. Coming also to NTA technology a little bit, we do standardization on a GMP level. There are protocols for how to determine standard derivatives and errors, and for standards and for the sample. So this is we offer that, and we do this in close connection with the customer. So basically tailoring the GMP, the IQ, OQ, PQ together with the customer, so they really get the result they want. Basically, once you want to, for example, we also assess agglomeration. Basically, how much percentage of your sample is agglomerated, and there are a few techniques out which can actually measure that. And so I think there's, in terms of stability, but please correct me if I'm wrong, for vaccines and everything. So once you have unknown samples in there, which are seeds for agglomeration, so they basically the reason why the thing agglomerates at some point. So perhaps agglomeration studies or the quantification of agglomerates versus the original product gives you a real number. Again, it has to be practical. So a measurement can cost so much, so you have to keep the costs down. Yeah.
You know, this is quite a good point. I mean, on NTA, certain standards have been done, but when you go to adopting your principle, when you go to more complex measurement or where you have interference, you need to account for your interference. And one of my questions to your talk is, how do you account for interference if you do gold or you do iron oxide in your system where you excite with a specific wavelength, and replacing laser for actually doing fluorescence to remove or to shift the absorbance, it's quite expensive because lasers, okay, don't come cheap. But also the other aspect that I've seen from some of the discussion we had with the MANTA is the fact when you have 3 lasers, you have the aspect that when you put 3 analytes into it, you have an overlapping on the signal. How do you take into account the fact that you don't have the so-called encroaching on the actual biological?
I guess so everybody can see me, so nobody has to wrench the head. Basically, once you do concentration, whenever you do concentration, it's key that you calibrate, and we don't make a fuss out of it. It is very important to calibrate, and once you measure gold, it's a different calibrator rather than polystyrene. It's based on refractive index, and this is key. If it comes to complex mixtures, we can scale with intensity. So we have the scatter plots. So we know the individual subpopulations, and we can really see the differences. And it's basically also what has been discussed with the runoff. Basically, you do not see so many of the small particles, rather to the large ones. More than 4 years, we've came out with an algorithm where we actually do not see this runoff. And again, for different types of materials, the better you calibrate with a standard, the better your result is. When you have mixtures, you also can calibrate that. This is not an issue. So each laser has been calibrated, so we have different so we have a whole calibration set. So a whole calibration table for fluorescent mode for each wavelength of a laser and for each fluorescent mode. So this has to be taken into account. Once you want to measure concentration, you have to take into account all that.
I have one comment to what you just said. So I totally get it's fair that you need to calibrate with a particle that's got the same refractive index in your sample. How do you deal with unknown sample? How do you deal with blind samples, actually? How do you calibrate it? Whether it's a mixture of gold plus latex, for instance, how do you do it?
It's because we just use a fixed camera setting, for example, and we have different intensities. And based on the intensities and the camera settings, we can actually derive the refractive index. So we can trace back what's actually close to the refractive index.
Right. Would that change if you're calibrating with a gold particle, and if you're calibrating the same with a latex or polystyrene bead? Would that be different?
Again, we are using then the intensity plots. So
Right.
we plot the intensity versus size, so we get the different subpopulations.
Okay.
And so we can use different calibration factors for the different populations.
Right.
Was this close?
Yeah,
to
that
the
makes
answer?
sense. Thanks.
And let's then dwell with the also question that has been debated across the different session, EVs and characterization of EVs, because this is becoming a need as well. And you have addressed in your presentation, Hans, on the new products and the new launch, and also Clemens, you have. I'd like to open a kind of a round robin discussion around how we can better provide data on this, because the information and the discussion is very hot at the moment, and this decision on the acquisition of a machine compared to another one determines not only a strategical choice, but also the ability to measure EVs instead of budding, apoptotic bodies, or fragments. What do you foresee in the pipeline in terms of having to use one of your words, a calibration or a control, a means of controlling from that side? I'll open the question for everybody. Hans.
It goes back to, if you're going to be doing biomarker development, for instance, if you have 15 laboratories all analysing samples, they have to use the same framework. So if you get 15 different instruments, that all give you different numbers for the same sample. You're just never going to get anywhere. And so you have to go back to what is the real number. And I'm sorry, but it's not a democracy. I mean, you know, we like a political system to be a democracy, but you get 3 different instruments, giving you 3 different answers. Which one do you pick? And it doesn't work.
No,
You've got to pick the one that works and not have the ones that don't work.
Obviously, you go to imaging techniques where you see them as the reference on your visual and volume.
But we would typically go to a lab, give a TRPS sample data, and they say, oh, can you explain why that's different than my zetasizer? And my standard, you better go and ask Melvin. Because our answer is the right answer, because everything's totally calibrated routinely. And the DLS is just not quite a random number generated, but on a complex sample it actually is. So for exosomes, for instance, DLS is utterly inappropriate. And you still get people submitting it. So you have to go back to finding out the real measurement and comparing that with calibration. That's a good place to start.
I think this is what I'm actually encouraging, the aspect of providing information that is as accurate as possible with the machine, but also having protocols in place that are reproducible, because some of the protocols, they're not exactly reproducible in terms of from the isolation, from the purity of the sample. Yeah. As we've seen in the other session, the purity of the EVs determine the effectiveness that then they can be used in a clinical settings. Is just not only measure it, it's also to measure the right fraction.
Well, the automatic fractionator is a step change for that. I mean, you went from ultracentrifugation or precipitation before that. Ultracentrifugation is a bit better. The manual set columns were a bit better. And now you've got the automated fractionation, which you now very, very precisely pick the fraction you want. So we're doing this work with the Mayo Clinic, and they're actually sub-fractionating and analysing with mass spec to then go back and say, well, for our big study, which fraction do we want? So everyone will pick, let's say, 0.5 to 2, for instance, and everything's done the same way. So that's highly repeatable. But then the measurement has to be based on something real. So the same when you've got mass spec analysis, which is part of it, and that's not that trivial either.
No, absolutely.
Probably harder than size measurement, to be honest, actually. The size measurement side of it's actually pretty simple, I think.
Compared to mass spec, yes. Yeah. And Jeff, from the side of expanding to creation of ISO standard, do you foresee working towards ISO standards in the direct specifics, especially for the new technologies?
So you can have an ISO standard for something utterly useless. And you do this all the time. Seriously, I'm not joking about this. There are ISO standards for Africa for things that are useless. So you standardise something that's useless. I'm sorry, what did you just achieve? And the answer is actually not much. So I can see the need for some ISO standards, but you've still got to get the basic measurement right. So there's ISO standards for DLS for Africa, and you push the same button 25 times. The data's still subject to huge errors. because a tiny little change in the refractive index or the temperature or the viscosity, you can't control those variables. So it's inherently the wrong technique. And it doesn't matter how much you standardise it, you'll never ever get to the right technique for that.
Sure, but that's where the full aspect of sizing has to be done through education
Sure.
of the scientific community.
But
So
it's not complicated. It's been made complicated. It actually is quite simple. And everybody's just getting this hugely complicated mindset that the question was, how do you validate it? I mean, and then what's the answer? I mean, you could talk about it for 5 or 6 days. But actually, there are techniques that are quite simple, quite calibrated, but they're not widely adopted for whatever reason, mainly historical uses. Or there's an ISO standard for DLS, or it's in the pharmacopoeia, or the FDA's got web pages about DLS, so then people just keep using it. And we're not going to get anywhere as a field until they stop. I'm sorry, it's just going to go around. Look, we've been here for this is the 11th Nano CLINAM conference. I'm sorry, what did they achieve in 11 years?
I think the fact that there is a workshop in sizing and concentration is already a step forward.
11 years later. It was
I
solved.
know.
It was solved 8
But
years ago.
alternative weren't. How many companies have been brought into the market? Only in the past 3 years, there's been a proliferation of techniques and instrumentation they came to compete against.
Melvin started, Nanosite started in 05, and
Yep.
we started in 09. Particle have been around for long, I don't know how long, but so it's not the last 3 years. I mean,
No, but I
it's
mean,
older.
in terms of, having a consolidated ground where to stand against the DLS, where data is generated,
Yeah.
there's groups that are taking up your machines and they're actually building
Yeah.
libraries of reliable data. Data is shared because now we have an open access commitment as a scientific community. So this is what I'm saying. I'm saying is an education over the process of, replacing, refining measurement, or completely changing radically the methodology for analytical sizing.
But I think there's a real place for NCL and NIST and others to educate the community, but they don't. They're too scared to. I don't know what it is. I go to all these conferences.
We're not
I've been-
scared,
I've been
we're
going to
here.
conference for 10 years. 10 years ago, I was told that DLS data wasn't very good at a measurement conference. 10
Yeah,
years
but
ago.
I mean,
And what's
Hans,
happened? Yeah.
similarly to this one,
Yeah.
even MTT for in vitro toxicology
Yeah.
is being proven that is not a reliable test.
Right.
But how many papers you still see? I mean,
Correct.
if you look in the block compartment for the cascade for assays,
Yeah.
they are fit for purpose in specific areas.
That's
So
right.
therefore, you need to take with a caveat of where
Yeah.
applicable.
I think that's very correct, and that's not the case, General. I agree with you. So when you're measuring something that's quite monodisperse, and Andreas was here this morning with his monodisperse set, then DLS is actually quite appropriate. But most of the users do not understand that. Your average PhD student in a chemistry laboratory is absolutely not told that.
So now we're coming close to the end, and I'll pull out my wild card. And this is a question for all of you. What in the situation as Jeff presented of complex media, where you have albumin, where you have fibrinogen, where you have complex biological entities that are actually dirty in your machine. So this is what it comes to the real measurement, if we go down the real measurement line. So any last thoughts on this? Because this is a work in progress.
Well, the talk that
Clemens?
with the dark field microscopy and all that, I mean,
Oh, yeah.
highly complex, but it
Martin.
elucidated out the data. But
Yeah.
it's not something that everyone's going to do routinely, but for research, I mean, it was very clear, but it's hugely complex. I mean, you're not going to just apply that routinely.
Absolutely. I mean, the technique that is shown, they are fantastic. And if you couple them with elemental analysis on the particle that he has done, you have an irreplaceable tools because you can pinpoint where the particle are. And this is where the evidence is based. I have some issues with Martin, but I'll discuss them some with him, because when you use H&E, H&E is a gold-based assay. So actually it's dirty in your sample when you measure gold. But that's something we found ourselves when we were doing H&E and we're looking in refractance and we say, whoa, shiny. Then you put gold and you see the same things. and then you go to the supplier and you see that there is gold. But that's
Adriel, yes, Luigi from Jersey. I miss. I had to be in another room, so I miss all the presentation in the afternoon. But if I can go to what Adriele was saying about how you're going to measure size of your particle in a complex media, let's say in plasma, in the presence of protein, really depends material to material. And in our experience, we are used with especially metallic nanoparticle, high-dense nanoparticle, and we have had a lot of success with different techniques presented some this morning, like centrifugal particle sedimentation, of a very expensive one, analytical ultracentrifugation. But apart from that, that's, I would say, what is critical from my point of view here, that would be to have a proper system of 2-3 different types of particles that are relevant for this community, and then start checking in control conditions. And that's where, and we have done these exercises ourselves for other type of simpler material in the past, because more gold and silver. And then we can start to confront the different technologies. That would be my way, because at Jersey we are agnostic in a sense that we use every technique that's available that fits for purpose, for example. But we like to check the performances of the others. And maybe one technique that is good for one material is not good for another. And I know exactly example of this. And we have also published some of these concept papers in the past for simpler material.
To pick up the discussion about complex samples, I think in particle analysis, it's important to gain a priori knowledge of the sample. I mean, in analytical chemistry, it's like you have a question like, I'm looking for gold, so I look for gold. In particles, you look for size and for concentration, and it's really dependent on the techniques of how the data is generated. So in terms of DLS or nanoparticle tracking, it's light scattering, and this is traces back to refractive index. So at least if you, for example, can feed a little bit of refractive index back to it, this helps. And so in terms of extracellular vesicles or biologicals, we know a little bit about refractive index, so it's not super high, such as for gold or for inorganic. So we have some estimates, and if you take the average, so you're quite close. So it's also a question: are we talking about 1% error, 2%, 5, 10, or 20% of error, which is acceptable? And the higher your constraints are, the more you have to treat your data, or you have to take care about the measurement of the samples. For extracellular vesicles, so a recommendation, if we come to a discussion, it is like, first check the instrument, basically check it with a standard you can really derive back. So like polystyrene, you can measure that. You get a mass concentration, you have the mass concentration, you trace back to the number concentration. This is what you can trace back. Then, so then you know that your instrument is actually working. And then we have in touch with a lot of companies for actually biological standards, such as liposomes. So they are quite similar in the properties to the EVs, and actually EV producers or EV isolators. So there's a lot of knowledge we are gaining at the moment, and this is what I can contribute to this discussion about the standards used for detection. My question back is also, why is an EV? Is it simply a empty vesicle, or do you have something on the surface? So should the surface and the surface proteins also be part of a kind of standardization? So that's the next question. So how specific you would like to be your standard actually?
Okay, unfortunately, as I check time, we have to wrap up, and I'd like to thank you, the speaker, and the audience, to come twice to the room. and walk all the distance. I appreciate everybody's contribution. Jeff and I are going to try to wrap everything, and what we have, as we have expressed our intention, is to actually create some material that is going to be up for consultation. And the idea is to inform the community as much as we can in every direction. So we value your input. So if we don't have your contact detail, or if you are particularly interested in some aspect, please do look for us, or we can make a further arrangement to continue this discussion, because this has been very valuable for all of us, even from the fact that Jeff has just come back from a US conference in ACS where he had the same topic for discussion. So we can compare the aspects that we discussed this. So I personally like to thank you. Jeff, I think you second me on this. And thank you very much. So enjoy the coffee.